Hello,
Hello.
I'm Erin Regan, and we're doing Chapter 7 for Microbial Regulatory Systems.
And I'm Julie Lee. I'm going to be doing the concept map.
Okay, so part 1 of this chapter is about DNA binding proteins and transcriptional regulation. So, for DNA binding proteins, bacterial and archaeal promoters have distinct nucleotide sequences which are bound by DNA binding proteins. And those proteins allow RNA polymerase to bind, and subsequently transcription can then occur. Um, the proteins interact with nucleic acids. The repeats can become inverted, which means the nucleotide sequence is followed downstream by an inverted complement. And there are frequently specific binding sites for regulatory proteins. The DNA binding proteins are often homodimeric, which means they're identical. They possess a domain, which is a region with specific structure and function that binds to one inverted repeat. And many different DNA binding proteins from bacteria contain helix-turn-helix domains, such as the LAC and TRP repressors in E. coli. For transcription factors and effectors, more transcription leads to more mRNA for translation and more protein product is then formed. So for the mechanisms of transcription factors, proteins control the rate of transcription by binding to specific DNA. An activator protein turns on transcription by binding DNA and recruiting RNA polymerase or the sigma factor to the promoter region. And repressor proteins turn off expression and bind an operator region of DNA downstream of the promoter. Effectors are small molecules that control the binding of activators and repressors. Typically, cell metabolites such as substrates and products or structural analogs like tryptophan or lactose. Allosteric proteins, their conformation is altered when effector molecules bind to it. Since transcription factors are allosteric, conformational change determines whether transcription factors can bind to the DNA. Inducers turn on transcription and co-repressors turn off transcription. In repression and activation, we can have enzyme repression and induction. In enzyme repression, there's the tryptophan operon, which prevents the synthesis of an enzyme unless the product is absent. And then the excess of product decreases enzyme synthesis. So it's widespread as control for production of amino acids and typically affects biosynthetic or anabolic enzymes. An example is the tryptophan operon. E. coli has the ability to make tryptophan, but can also absorb it from its environment. The tryptophan operon is negatively regulated by the TRP repressor. When excess tryptophan is present, it binds to the repressor protein and activates it, which then binds to the operator sequence and prevents RNA polymerase from transcribing the operon. And then when there's no tryptophan, the repressor is inactive and the operon is transcribed and translated to make tryptophan. In repression and activation, again, we can have enzyme induction, which is the opposite of repression, production of an enzyme in response to presence of a substrate. So that typically affects degradative or catabolic enzymes, such as in the LAC operon. This process ensures that enzymes are synthesized only when they're needed. When there is no lactose, the repressor protein binds to the operator sequence, which prevents RNA polymerase from transcribing the operon. And then when allolactose is present, which is the inducer, it binds the repressor and deactivates it, allowing transcription to proceed. And this is just a graphic showing the LAC operon. Mechanisms of activation. Some operons are transcribed only if the activator protein is first bound to DNA. Promoter sequences are poor matches to consensus promoter sequence, thus only weakly bind the RNA polymerase. In positive control, the regulator protein facilitates transcription, such as in the maltose operon, where the operon is transcribed only after the regulator protein, which is activated by the maltose, binds to the activator binding site, which is upstream from the promoter, as seen in this figure.
Hello, my name is Margaret, and I'm going to be reading off the slides for chapter 7.
I am Julie Lee, and I'm doing the concept map.
This is 7.3 for sorry, reparation I can't say that word right now. Repression and activation. Operons versus regulations. Genes for maltose are spread out over the chromosomes in several operons. Am I saying that right? Operons. Each operons have an activator binding site. Maltose activated protein control transcriptions of more than one operon. Multiple operons controlled by the same regulatory protein are called a reglon. Examples are maltose and maltose reglons. Reglons are reglons also exist for negatively controlled systems. Multiple control features common in many operons are reglons. 7.4 archaeal transcriptions regulation more closely resembles control by bacteria and eukarya. Archaea use both positive and negative control. Receptor proteins in archaea either block RNA polymerase binding or block binding of TAT, a binding protein, and transcription factor B, which are required for archaeal RNA polymerase to bind to its promoter. Bacteria and archaeal promoters characterized by distinct nucleotide sequences recognized and bound by DNA binding proteins allow RNA polymerase to bind transcriptions to occur. For 7.5, two-component regulatory systems. Prokaryotes regulate cellular metabolism in response to environmental fluctuations. In most cases, external signals may be detected by sensor and transmitted to regulatory machinery, signal transduction. Two-component regulatory systems containing two parts. Sensor kinesis, which detects environmental signal, and autophotolites at specific histone residue. Response regulators, which are found in the cytoplasm, are DNA binding protein that regulates transcriptions, receives phosphates from sensor kinesis. Also has a feedback loop, which terminates responses and uses phosphate that removes phosphates from response. And then this is an image of the control of gene expression by a two-component regulatory system. 7.5, two-component regulatory systems. Examples of two-component regulatory systems. Almost 50 different two-component systems in E. coli. Examples include osmotic pressure response. You can go to the book or in the presentation, I believe there's a picture. And then here's a list of examples. OMPC and OMPF genes are controlled by environment, osmolarity, ENVZ sensor, Hendine, kinase detects osmotic pressure changes. Autotrophosphates are give phosphates to transcription regulatory proteins, OMPR, which bind upstream from OMPF and activates transcriptions of its underlow um, osmolarity. OMPR only activates the OMPC under high high osmolarity. And for 7.6, regulatory of chemotaxis. Chemotaxis, it moves towards attractants away from repellents. Response to temporal gradients. Examples of this would be Pseudomonas aeruginosa. Bacteria use modified two-component systems to sense temporal changes in attractants or repellents and regulate flagellar rotations. Thus regulates activity of pre-existing proteins instead of modifying transcriptions of genes. I think that's the 5 minutes.
Okay, hi guys. I'm Ajong Nguyen, and I'm reading the slides.
I am Julie Lee, and I'm doing the concept map right now.
Okay, so regulation of chemotaxis. The response to signal depends on the signal cascade of multiple proteins. The path is the chemical attractant or the repellent goes to MCPs, then the sensor kinase, CHEA and CHEW, then goes to the response regulator, which is CHEY. Then it goes to the control of the flagellar rotation. The chemical attractant is a nutrient such as glucose. The methyl-accepting chemotaxis proteins are sensory proteins that sense chemicals such as attractants or repellents and interact with cytoplasmic sensors, kinases. The chemoreceptors are clusters of thousands of MCPs. E. coli has 4 transmembrane chemoreceptors. MCP binding of attractant or repellent triggers interactions with CHA, which is the sensor kinase, and CHEW, coupling protein. Phosphate transferred to CHEY, the response regulator that controls the flagellar rotation. Let me actually minimize this. The regulation of chemotaxis, again, controlling the flagellar rotation. CHEY response regulator governs the direction of the rotation of the flagella. Counterclockwise, the bacterial run, which is when they swim smoothly towards the signal. Clockwise, the bacterial tumble, which move randomly and changes directions. When MCPs bind repellent, CHEYP is phosphorylated, interacts with the flagellar motor to induce clockwise rotation and tumbling. When NCPs bind to attractants, the unphosphorylated CHEY does not want to bind to the flagellar motor, resulting into a counterclockwise rotation and run. Adaptation, their adaptation is to stop responding and reset the sensory system. Feedback loop resets the system. It relies on the response regulator, CHEB, and involves methylation of MCPs. Methylation stops responses to attractants and increases the response to repellents. Other taxes are CHE proteins also play a role in other taxes. Phototaxis, which is movement towards any light. Light sensors replace the MCPs. Then aerotaxis are movement towards oxygen. Redox proteins monitors the oxygen levels. Sensors interact with cytoplasmic CHE proteins, leading to the runs and tumbles. Prokaryotes can communicate through production of small extracellular molecules, small peptides or non-peptide organics. Accumulation leads to coordinated group behavior, such as biofilm formation. Quorum sensing is regulatory mechanisms by which bacteria and some archaea assess their population density. Quorum sensing regulates a lot of processing, including bioluminescence and biofilm formation. The mechanism of quorum sensing ensures sufficient numbers of cells are present before initiating activities that require a certain cell density to be effective, such as toxic production by pathogenic bacteria. Each species produces a specific autoinducer signaling molecule. They diffuse freely across the cell envelope outside. After reaching a threshold of concentration, they diffuse right back into the cells. They reach high concentrations inside the cells only if many cells are nearby and make the same autoinducer. They bind to specific activator proteins or sensor kinase, triggering transcription of specific genes. Examples is the Lux operon, which encodes the bioluminescence or luciferase in Olivibrio fishery. The fluorines induced only when a quota is met. This is an image of quorum sensing. Quorum sensing contributes to virulence in some strains, such as E. coli. The Shiga toxin producing strain acquired pathogen island from Shigella, or it produces AHL that induces virulence genes, which is a type of autoinducer molecule. Epinephrine plus norepinephrine produced by a host intestinal cells, and AI3 bind to sensor kinase in cytoplasmic membrane. They activate two transcriptional activators, thus activating motility, toxin secretion, and production of lesion-forming proteins. The interspecies recognition. E. coli recognizes self-produced autoinducer signal produced by other microbes such as salmonella or the human stress hormone epinephrine or norepinephrine.
My name is Tamia McWhorter, and I'm going to start reading from slide 26.
I'm Julie Lee. I'm doing the concept map right now.
Perfect. The lac operon. Lactose operon and maltose regulon respond to a higher level regulatory mechanisms called global controls. The catabolite repression is an example of global control, and it controls the use of carbon sources if more than one is present. Glucose is always used first. Synthesis of unrelated catabolic enzymes, an example is lactose operon and maltose regulon, is repressed if glucose is present in growth medium, also called the glucose effect. The lactose operon is repressed even though lactose, the inducer, is present, and this ensures that the best carbon and energy source is used first. The catabolite repression is a form of global control, and together with being under negative control of a lac repressor, the lac operon is also under global control. The catabolite repression leads to deoxic growth, which is observed experimentally, and two exponential growth phases if two energy sources are available to a microbial cell. So better energy sources, glucose, is degradation ensures production of the most ATPs, and it is consumed first, which stops the growth. After a lag or a delay, growth resumes and is powered by the second energy source, which is usually lactose. During the lag phase, the cell synthesizes all the proteins from the lactose operon to prepare for its degradation and the ATP production. And this is a graph illustrating this process. The lac operon, the cyclic AMP and the cyclic AMP receptor protein. In catabolite repression, transcription is controlled by the cyclic AMP receptor protein and the activator protein, and it's a form of activation, which is kind of oxymoron. The CRP is allosteric, and then it binds to DNA only if it has first bound cyclic adenosine monophosphate, or the cyclic AMP or CAMP. The regulatory nucleotide synthesized by the adenylate cyclase. The cyclic AMP and cyclic AMP receptor protein is for lac genes to be transcribed, and the cyclic AMP level must be high enough for CPR protein to bind, and the CRP binding site, which is the positive control. The lactose or another inducer must be present to prevent lactose repressor, LACL, from binding over the negative control. So there's both a positive and a negative control. When the lactose is present and the glucose is absent, there's enough CMP present in the cell to bind to the CRP protein, which becomes active and binds to CRP activated site, which is similar to activated binding site, and recruits the RNA polymers to bind to a promoter. It begins transcription of lac operon, since the lac repressor is now bound by lactose, which is an inducer, and change shape and not bound to DNA. When both lactose and glucose is present, there is not enough CMP in the cell to activate the CRP protein, and thus the lac operon is not transcribed. So the glucose inhibits the synthesis of cyclic AMP from ATP using adenylate cyclase enzyme and stimulates transport of the CMAP out of the cell. And this shows the process. The stress survival pathways improves chances of survival under harsh conditions, and this is all under the stringent and general stress responses, which are also examples of global control. The mechanism of the stringent response is widely distributed regulatory mechanism to survive nutrient deprivation, environment stress, and antibiotics, and after cells convert to persister cells. It shuts down macromolecule synthesis proteins and activates pathways to improve survivability. Production of tRNA and rRNA and protein stops to avoid production and accumulation of a non-properly folded proteins, so no new ribosomes are produced. Synthesis of certain new amino acids are not available any longer from environment, as well as proteins begins on existing ribosomes. After a while, production of new ribosomes begins again, but at a lower rate, and the response is triggered by the accumulation of special molecules, the alloremones.